What's up, baby bio penguins? So I promised my students that I would do a Unit 7 live, and although there's probably going to be no one showing up because, well, we're currently in my planning period, so it's like 1 o'clock at school, so I know there's no one going to show up, but I promised them I would do it, so here is the crazy time that I'm doing it, even though no one's going to be able to show up. And I'm actually in a different spot than normal because, well, I'm back at school. So I promised that I would do a Unit 7, which for my students is actually Unit 5, a Unit 5 live. And so I'm going to go ahead and just do it. I'm going to record on IGTV, so don't worry, it's going to be saved. But I did want to promise them that I would go through it, so I'm going to just go through my normal thing where I have my little PowerPoint. And then I'm going to do a long free response question and a short free response question. Yes, I know I should be grading, but I promised them since they have their test tomorrow that I would do the live for them. So what is Unit 7? So Unit 7, and for my students, that's why it says Unit 5, is because my students is Unit 5. It's going to be Hardy-Weinberg, and I do already have a video that goes over the math for Hardy-Weinberg. So there's a quick thing. I'm just going to go over what Hardy-Weinberg is and what it's about. Talk about the biological species concept. Talk about the different reproductive barriers. And of course, just talk about selection in general. So first thing, if you have not watched the 5 Fingers of Evolution, it is a TED Ed video from, I believe it's Paul Anderson that does it, the gentleman that's behind Boson Bio. He came up with the 5 Fingers of Evolution. And this is a perfect way to remember it. And the reason why is because it actually just works out so good and you can really remember it. So what he says is, okay, well, every finger on your hand represents a component or a condition to Hardy-Weinberg. So overall, Hardy-Weinberg says that a population is not going to evolve if these conditions are met. Okay, so as we see, we have our 5 fingers. The pinky finger is going to represent that we need to have an extremely large population size, okay? So the larger your population size, the less that small random changes like genetic drift are going to affect your population. And so we need there to be an extremely large population size so that these small random situations that happen aren't going to change our allele frequency, because that's what's happening with evolution. Is evolution means that there is a change in either the allele frequency or the genotypic frequency. So we need to prove, okay, this is not happening. And so in order for it to not happen, we definitely need to have a very large population size. Okay. The second thing that we have to have, and if you notice, I'm actually wearing my rings today. My students know that I forget my rings on the daily. So the ring finger represents mating, right? So we need to have random mating. Okay, there cannot be one trait that is favorable. We can't have intersexual or intrasexual selection. We have to have random mating. Okay, it has to be random, so there's no potential for one trait to be more favorable or one trait to be less favorable in terms of mating. And so that kind of goes back to the good gene hypothesis that we talk about with the blue-footed boobies. Which, if you don't know about blue-footed boobies, they're like one of my faves. They do a little beautiful dance, and they show off their beautiful little feet, and that dance is actually a mating ritual, so that they can say, oh, you know, I've got the good feet, I've got the good genes, you want to mate with me. And it's just part of intersexual selection. So with it, we need to make sure, of course, we need to have random mating. You can't mate with that blue-footed booby because, well, he's got the pretty feet. Okay. So another thing is the middle finger represents no mutations. Every time that there's a mutation, it means that there's a new allele. There's a new characteristic, okay? Even though it's all the same trait, we're talking about a new variation to that trait, which if you add a new variation, you're automatically going to change the allele frequency because we've got a whole new trait in there, a whole new variation to that trait in there. Okay. The next thing is that we can have no gene flow. Okay. So your pointer finger kind of represents going in and out of a population. Okay. If there's individuals moving into your population or individuals moving out of the population, that's automatically going to change your allele frequency or your genotypic frequency. And last but not least, natural selection. You can't have natural selection. Nothing can be more favorable or less favorable in a population if we're wanting the evolution not to happen. Now again, these 5 conditions mean evolution did not occur. So extremely large population size, random mating, no mutations, no gene flow, and no natural selection. I always tell my kids, like, no, no, no. It's kind of mean, but it's okay. But if you've not watched that 5 Fingers of Evolution, is just search it on YouTube. Super easy, 5 Fingers of Evolution, and it perfectly explains it, plus gives you a real world example about redheaded individuals at the pole versus the equator. It also gives you a little bit of information about skin tone, which is kind of cool and interesting. So the next thing about Hardy-Weinberg is that there is a beautiful formula. Yes, I know we love math. And of course, I think this may be from Miss Kim Foglia, because there's all these penguins on it, which if you know anything about me, of course, penguins are my fave because, well, they're dressed for success. But anyway, so the equation we have here is p squared plus 2pq plus q squared equals 1. Okay, so P represents your dominant allele. If you have trouble remembering that, a P is just an upside down D. So if I have 2 of my dominant alleles, that means I'm homozygous dominant. So P squared represents homozygous dominant. Q represents your recessive allele, and kind of the tail of a Q looks like an R. And I can help you remember recessive. And so if you have 2 of the recessive alleles, then that's going to make you homozygous recessive. So the Q squared represents the frequency of the homozygous recessive. And then 2pq, you have 1 dominant and 1 recessive allele. So that would be your heterozygous individuals. Okay. And I'm seeing there's a lot of individuals coming in. So just FYI, I am saving this on IGTV. I promised my students I would do a live. So I'm holding true to my promise, and this will be posted, I promise. Anyway, so with the Hardy-Weinberg, that's how we're going to calculate this population. And we're going to determine whether the allele frequencies are changing. If your P changes from one generation to the next, you are not in Hardy-Weinberg equilibrium, and your population is evolving. So that's one of the things we're looking for with this math is, did the population evolve? And there could be, I don't know, some type of chi-square on this, where they give you the P and the Q before, and maybe they are having you, what do you expect to happen? Well, you expect it to stay the same, and does it vary enough to prove that we're not in Hardy-Weinberg? And so there's a whole mess of things that they could ask you, okay? But so when you're doing the math for these, there's 2 different ways to do the math. And that's the problem with this, okay, is you have to know which math to do. Because if you don't do the right math, you're not going to get the right problem, the answer, okay? And this came up a couple years ago when we had our numeric responses. And every year, and just FYI, teachers fall into the same traps as students do. So if you fall into a trap that the test writers write sometimes, don't worry. We do the same thing. So there's a question, and it gives you out the different the different genotypic frequencies of this population. Like, it tells you how many everyone is, and it says before or after, and they want to know, well, calculate this one trait. And you sit there and you go, oh, easy peasy, and they calculate it. Well, they calculated it using the wrong formula. And so because they calculated with the wrong formula, they got the wrong answer. It was a numeric response. So of course, you didn't know that you were wrong. So there's 2 ways to do it. If the question gives you the number of homozygous dominant, heterozygous, and homozygous recessive, if they tell you all 3 of those numbers, you know your P squared, you know your 2pq, and you know your Q squared. You have to calculate by counting your alleles. Okay, so individuals that are homozygous dominant have 2 dominant alleles. So you multiply the number of homozygous dominants times 2, and then your heterozygous has 1 dominant and 1 recessive. And so you'll add in the number of heterozygotes because of the fact that you only multiply those by 1. And that tells you the number of dominant alleles, and then you divide by the total to get your P and your Q. Well, your P. And then you do the same thing looking at homozygous recessive times 2, plus your heterozygous, which would give you your number of recessive alleles, okay? So that's the first way to do it. And the second way is to use, of course, the P squared plus 2pq plus Q squared formula, okay? And if you use that formula of the P squared plus 2pq plus Q squared formula, you always want to start with Q squared, okay? Because of the fact that if I have a dominant phenotype, I could be P squared or 2pq. So you always want to start with Q squared for your math so that you automatically know that you're looking at only one variable. Plus, once you have Q squared, square root of that, find the Q. Once you got the Q, you then find the P. Once you find the P, you find the P squared, and then you just do 2PQ. So it's kind of easier math. And of course, I've done a video already. There's a live on this, on the math for Hardy-Weinberg. So, and I worked out, I think, 3 or 4 different problems. So definitely check out the IGTV if you're needing some help on the math problems for Hardy-Weinberg, because I did examples of all the different types of ways that you could see a Hardy-Weinberg problem. And I did those, oh gosh, I did like in September or October. Like, it's been a while ago. One of my earlier videos. Wasn't a good one, I promise. Anyway, so biological species concept. This is, of course, the ability, okay, what determines that 2 individuals are in the same, or 2 individuals are from the same species? Well, can they interbreed? So can they, when they meet, can they breed with one each other? And if they breed, is that offspring viable and fertile? If the answer to that is yes, then they're considered the same species in terms of the biological species concept. This doesn't apply to, hi Melanie, this doesn't apply to a lot of the other things. Like if we're looking at fossils, I can't tell if a fossil was able to mate with another individual. They're fossils. But so for anything that's living, we can apply this. And the same thing, we can't apply it to something that's asexual because, well, you can't see what happens when it mates with another because, well, it's asexual. But so for most things, we're going to look at the biological species concept, which is, can they breed, interbreed? If they can, do they produce fertile, viable offspring? So when you think about this, there are barriers that stop species that aren't the same species from mating together. And so a lot of students love going through this. This is one of their favorite parts, is the prezygotic and the postzygotic barriers. So prezygotic, take the word apart, right? Pre means before. And of course, we know the zygote is what's formed when the sperm and the egg come together. So that's your prezygote. And so prezygotic is something that happens before the zygote is formed. And so first, let's think about those barriers. So the first one we have is, of course, here, a habitat isolation. We've got 2 different snakes. This snake lives in the water. This snake lives on land. They live in 2 different places. They're probably not going to come in contact with each other. And so since they don't come in contact with each other, they're probably not going to mate. And so that would be habitat isolation. They're actually isolated based on their habitat. Another type of habitat isolation is geographic isolation. Just means they're in 2 different geographies. There could be a geographical barrier, like a mountain range that separates them. And so because of that, we could see that, okay, these are not going to mate with each other. The next one that we have is temporal isolation. And so I cannot remember which is which, but one of them is an eastern skunk and one of them is a western skunk. I think this is eastern. I think this is western. It truthfully doesn't matter. But one of them is going to mate in the summertime while the other one mates in the wintertime. And so because of the fact that they mate at 2 different times of the year, they're not going to mate with each other. And so we call that temporal isolation. And you may have seen temporal before when you looked at photosynthesis, when we talked about the cam plants. So remember the cam plants, they were like your cacti, and they would open up their stomata at night when it was nice and cool in order to bring in that carbon dioxide. And then they would collect all their sunlight, of course, during the day. And so they had a temporal separation in terms of when they were going through the parts of photosynthesis. So temporal kind of makes us think of time. So the temporal isolation is that they're isolated based on the time of the year that they're going to mate. They could mate at different times of the day, different times of the year. They could mate different years in general. And so because of that, they don't mate with each other. And next one we have is our behavioral isolation. Of course, look, it's my blue-footed boobies. Aren't they cute? Oh my gosh. Okay, sorry. Anyways, so behavioral isolation has to do with different behaviors. So maybe we're looking at a mating dance, right? The blue-footed booby does a mating dance, and with its mating dance, it lifts up its beautiful blue feet. And by lifting up those beautiful blue feet, it tells the mate, hey, you want to mate with me? I know you do. And so it's trying to attract a mate through that process. Same thing happens with like the song. So any type of mating ritual would be considered a behavioral isolation. If they have different rituals, they're not going to mate with each other. Okay. So now we've gone through all those. Let's pretend that they're in the same habitat. They mate at the same time. There's no different ritual. Let's say that they try to mate. Okay. Now, the next type of part of prezygotic could be mechanical isolation. They just don't fit together. There's anatomical, morphological differences that inhibit the two from mating. So in our example we see here, we've got these snail things. I don't think you can see the arrow, but I'm gonna get real close. So you see that there's an arrow here and an arrow there. Okay, so that arrow is where their reproductive parts are. And so since they don't line up, they can't mate. So that'd be mechanical isolation. And last but not least, thinking about external fertilization, there's sea urchins, right? These sea urchins lay their eggs along the water, right? The bottom of the water. And when they do that, the sperm then gets released out into the water. And so you don't want a sperm to fuse with an egg from two different species, right? So what we see happening is that there's different glycoproteins on the outside of that egg to inhibit the wrong sperm from fusing with the wrong egg. And so that is considered gametic isolation. And so here's our last one, gametic isolation. This would be our sea urchin, because of the fact that the sperm is not going to fuse with the egg, because they don't fuse together. And so those are all the prezygotic, the things that happen before you form the zygote. Okay, so now the zygote's formed. They mated, the sperm egg came together, the zygote was made, boom. Now we're talking about post-zygotic, post meaning after, right? So after that zygote is formed. And so with the post-zygotic mechanism, we're looking at three different things that could happen after the zygote is formed. So the first thing is reduced hybrid viability. Kind of hard to explain, but overall, there's just medical issues with it. It is not viable. And if you remember, we said that in order for them to be the same species, they had to be able to breed and produce fertile, viable offspring. They had to make healthy offspring. This lizard right here sometimes is born without a tail, and well, it's just not healthy. It's not viable. It's not going to survive for a long time. And two organisms aren't going to mate to produce an offspring that isn't viable, or at least evolution tells us that they're not going to. And so the next one we have is everyone's favorite example, the horse and the donkey when they mate. And I'm crazy. I've always called them a horky because I thought it was fun to say that. They're actually called a mule. So when a horse and a donkey mate, they make a mule. And so I cannot remember the chromosome number off the top of my head. Okay, but you got your donkey, you got your horse, and they make this beautiful little mule down here, right? And so the mule has an odd number of chromosomes. And so we know that if you have an odd number of chromosomes, the homologous pairs can't pair up during meiosis, which means that we're going to see an inability to make sperm or egg. And so because of that, they're considered sterile. And as we said before, they have to produce fertile, viable offspring. That mule is not fertile, it is sterile, so it is actually considered a post-zygotic barrier. We're not going to mate if my offspring can't continue to mate, okay? It's, again, an evolutionary thing to try to stop mating of things that aren't even going to continue producing. Hi, Elizabeth. And last but not least, we have our hybrid breakdown, okay? So hybrid breakdown, first generation, perfectly healthy, no issues, we're good, okay? But the second generation is when we start to become a little bit feeble. It's not as strong. It's a little more weak. And it starts to break down. And for those of you who just joined, I promised my students I would do a live. This is not a time that any logical person should be on Instagram because this is in the middle of the school day. But I am saving this, so don't worry. But I know you should all be in class right now. I just promised my students I would make the video for them. So last but not least, we need to think about speciation. Okay, and so with speciation, this has to do with the production or the making of different species. So when when one species becomes two different species. So there's two types of ways that this can happen: allopatric and sympatric. Allopatric has to do with that these individuals are apart from each other. Hi, duck. In which, since they're apart from each other, there's an absence of gene flow. Okay, gene flow, as we said before, was immigration, emigration. There's an absence of the alleles kind of intermingling within the gene pool. And so because of that, we see that if there's a mutation in one of the populations, that population could diverge and become its own different species that it no longer is able to mate with the first one. Sympatric means that they're in the same era. So there's some issue that happened that they stopped mating with each other. Maybe one of them choose the new tree. So for example, I think it was, there's these flies, and they used to be in the apple trees, and then the hawthorn tree was introduced, and because of the hawthorn tree was introduced, the flies preferred the hawthorn tree, and so you saw a habitat isolation because now they preferred two different habitats, so they no longer mated with each other. And so allopatric is when they are apart, and sympatric is when they are together in the same place. So selection, right? So we have natural selection, right? But what's actually happening with natural selection? Because organisms, the whole population is changing, right? So we kind of think about, well, how did the population change? Well, there's three things that could happen. The first thing is directional selection. It's moving in a direction. Okay, so back in the day, the peppered moths, they were a light color because of the fact that the trees were this light color, nice, beautiful speckle color. Then the Industrial Revolution happened, and there was all this dirt and smut and nastiness all up in the air, right? And it covered the trees. And so when it covered those trees, now these light-colored moths stood out when they landed on those darker trees. So now they were picked off and they were preyed on, right? So any of the individuals that had a mutation that allowed them to be darker could kind of blend into the tree, and then they were not as preyed upon. And so we saw an increase in the darker peppered moths. So over time, it was moved in a direction from a lighter color of moth to a darker color of the moth. Okay, and so that is considered directional selection. So we're going to move in a direction. So we move from one to the other. Okay, so here you see we have, this is looking at beetles, but you'll start out with a lighter color and they become a darker color. So we moved in a direction. And FYI, the little dotted is your original population. The solid line is after selection, and the red is showing like which one was selected against. So next we could have is disruptive selection. So example we can use here. Oh, wait, sorry. I flipped it too many times. Example we can use here is that there was a drought on Daphne Major where the finches were. And so there was a certain type of seed that was not surviving because of the drought. And so the birds that had the itty bitty little beak could get into the smaller parts, get the bugs, and they were perfectly fine to eat those, right? And then the big seeds that there were, only the birds that had the big, robust beaks could break those seeds. And those medium-beaked birds, they couldn't really get the bugs, and they couldn't really get the seeds. And so they were selected against. And so we saw disruptive selection. So we saw a decrease in the middle type beaks and an increase in the two extremes. And that's considered disruptive selection. So here you can see that. So here we're seeing an increase in our two extremes, and then a decrease in our the medium in this case. And last but not least, stabilizing selection. Good example for this is birth weights. So if the baby is born, like we're thinking like evolution, long time ago, right? Babies that were born too small were too small to survive. And so we found that, there was a stabilizing to kind of make sure that the weight was a little higher. Babies that were too big had trouble being born. And this, of course, before common medicine. And so they would die before they could be born. And so big babies were selected against. And so stabilizing selection kind of selected for that kind of middle range. So kind of think it to yourself or ask your parents, how much did you weigh when you were born? And I bet you were somewhere in that ballpark of 6 to 8. Now, modern medicine has helped us. And so there was a lot of individuals. I know that some of my students told me they weighed 2 pounds because they were little preemies, which is awesome. But my best friend had a preemie. She had her baby like 34 or 35 weeks. It was itty bitty. Anyways, but so due to common medicine and our normal, now we can actually have this taking place. And then we also see with the larger babies that we have C-sections and they could be born. So with our selection, natural selection overall with this is that we're looking at that there's some trait that's selected against. And so in this case, you can see that our allele frequency of our dominant was 0.3, and then our recessive was 0.7. And then after we went through natural selection, we see that the dominant, our P, went up to 0.6 and our recessive went down to 0.4. Okay, so with natural selection, this has to do with that some trait is more favorable. And because that trait is more favorable, those individuals leave more offspring. And because they leave more offspring, there is a higher chance of those individuals surviving and continuing to leave more and more offspring. And so over time, we see the increase in the favorable allele and a decrease in the unfavorable alleles. Okay. So I just remembered that I have to do office hours with a student. So I cannot go through the free responses right now, but I promise you I will come back and do a second live in which I go through the 2 free responses that I had planned. I've got 1 long and 1 short free response that I'll go through later. So I hope that this was helpful, and I will be back later for part 2 of my live. Bye, everyone.